it's going to be, it's not just because I'm increasing my production to reducing the amount that I have to water my plants, because I pay for water. We can't water our plants with our wells, so I pay a water tanker, right? So it's obvious to me looking at this that there are immediate ways that I'm also going to be able to increase the production I've got. So, Elaine, I'm the first one to take up your offer, okay?
Other things about um, getting the biology back into the soil. The plants will sustain that biology. Get the system set up and the system sustains itself. That soil keeps growing. So it's not.
I've seen that on my place here in California in the Central Valley. The aspect doesn't even need to be straight on. And I don't use fertilizers. I just alluded to, for some of us, it's a lot of work, and we're busy doing other things. We got families to provide for, and it comes. I'm doing that, and then you go out there and you can easily distribute it, and you can make a foliar spray of it. So, I mean, I don't know. I honestly don't know how I could have these techniques without using compost tea. And for me, it all really started with Elaine Ingham.
We've got a lot of questions to go through. So, Matt and Neil, might need you guys help with this.
Question.
Okay.
Do I double click on the question?
Yeah, the urgent question is a little hand icon, and I think it should allow you to have a question. If you have a question you want to ask over your microphone.
And that's very cool. But, now I see.
Okay, let's start with let's start with Gene Berry. Is there a way I can't click on, so hopefully you can do an environment. Matt, can you read out the rest of that?
I'm trying to find where you're at. I do not have that. Let's just go from I just don't have that. Let's, in the interest of time,
Question happening right now. We're going to start with the last person that asked a question. Matt, how about click on the first question you get?
are very important for certain kinds of plants. Not all
Okay, so I'm going
plants
to do a
are
sound check.
mycorrhizal,
Can
and
everybody
it's
hear me
actually
and hear the video? Type in
inhibitory. It's definitely
if you are hearing everything clear. If not, I'll try to adjust the sound. So let's see if anybody, because Alicia says she has problem hearing the video. Anybody else in the audience? Tell me if the audio is clear. Alicia, can you hear me okay? Or is it just the not clear? You are clear, the video sound is bad. Okay, let's try playing this. See, okay, you can hear me well. Let's try playing this and see if the sound gets better. I hope it will get better later in the video. So let's try this.
detrimental to things like the brassicas, the cole, the kale crops, the mustards, to become if you've got a plant that is strongly mycorrhizal, it can send out those mycorrhizal hyphae and force brassicas should never become mycorrhizal or, not really helpful for them and will actually suck nutrients out of the non-mycorrhizal plant and cause it to die. So our plants must be mycorrhizal. It benefits everything. Nature doesn't work that way.
Yeah, let me try to play this one more time. Let's see what happens to the video. And
it's recorded.
I have just did.
Blessed to hear all that. Neil, do you have a Saudi Arabian question?
No,
Like
no. I actually got.
Okay, how about now? Is the audio better? Sounds better. All right, let's go.
Got the link from Raleigh early,
Mm-hmm.
and I have purchased the 3-course package.
Nice.
But here's the reason why. I have a 90-acre farm in a really extreme place, and there were a couple parts in this presentation where I was like, I need to know how to do this, right?
Mm-hmm.
I've got 1,000 moringa trees. I have 800 ziziphus trees. I've got about 1,400 mesquites, and I know that if I can increase the production off of these, that it's going to help me push the case to implement this system that I'm not pioneering it, but I am the first one to do it in this area. And so I just spent $1,500, which is more than I normally spend on a given instance, but I have faith that this is going to pay for itself on the farm that I'm running here in Saudi Arabia, and that's why, honestly, that's what it is to me. To me, it's it's not just because I'm interested in soil, even though I am. It's that I know this is going to pay for itself through increasing my production, through reducing the amount that I have to water my plants, because I pay for water. We have a well that's 140 meters down, and it's toxic water. We can't water our plants with our well, so I pay a water tanker to bring water to my farm. The water is free, but the tanker costs money, right? So it's obvious to me, looking at this, that there are immediate ways where I'm going to reduce my expenses, and I have faith, after seeing what I've just seen, that I'm also going to be able to increase the production I've got. So, Elaine, I'm the first one to take up your offer, okay?
Great. Yeah, fantastic. Yep. So it's
Awesome.
you know, and one of the other things about getting the biology back into the soil, the plants will sustain that biology. You do not have to be adding compost every year or compost teas. You get the system set up and the system sustains itself. That soil keeps growing. So it's not the constant applications that you have to do with a chemical agricultural system.
I've seen that on my place here in California in the Central Valley. My soil's hit 140 degrees where it's not watered and it's in direct sun. The aspect doesn't even need to be straight on, and I don't use fertilizers. I just do chop and drop and use compost tea. I don't spread my compost around because it's too precious, like as you alluded to. For some of us, it's a lot of work, and we're busy doing other things. We got families to provide for, and the compost tea is just so easy. You can grab 6 buckets, and here, you take this. Boy, you're doing this, and I'm doing that, and then you go out there and you can easily distribute it, and you can make a foliar spray of it and do it spray it on the plants, and kids love that. or you could just put it directly on the soil. So, I mean, I honestly don't know how I could manage and get the yields I get with the amount of time I put in without these techniques, without using compost tea. And for me, it all really started with Elaine Ingham.
Cool. Okay, guys, now it's time to answer some questions because we've got a lot of questions to go through. So, Matt, Neil, I might need your guys' help. There's a lot of questions, and for some reason I can't click on the question box. So, Matt, can you help me out with this? Why don't we...
questions.
Okay.
Do I double-click on the question?
Yeah, double-click on the question, and then you can send it to all. And if you guys have an urgent question, there's like a little hand icon, and I think it should allow you to have a question pop up. So, whoa, I can actually unmute you guys if you have a question you want to ask over your microphone. Whoa. Well,
that might be how we have to do it. I'm looking at this, and I see where everyone's from, and that's very cool. But oh, now I see.
Okay, well
some people just put their place down where they live. All right, I get it now. All right, so I'm going to the bottom.
Yeah, let's start with Jean Berry. Is there a way this? I can't click on it, so hopefully you can. It starts with Jean Berry. Is there a way this process can help with soil remediation to an environment? Matt, can you read out the rest of that?
I'm trying to find where you're at. Jean Berry.
Yeah, 1:29 p.m.
I do not have that. Let's just go from I just don't have that. I have Carolyn Kirkwood at the bottom, and then I could go back up, but I do not see Berry.
Okay.
Let's, in the interest of time, I have Bremen, Stewart.
Yeah, if you guys, I'll go about this. If you have a question, type it in right now. We're going to start with the last person that asked a question. Matt, how about click on the first question you get, and we'll just keep going that way.
Okay, so how does your work match with Paul Stamets' research? That's a...
I've worked a lot with Paul. I think Paul is incredible. He focuses on mycorrhizal fungi, or fungi in general, but a lot of focus on mycorrhizal. And mycorrhizal fungi are very important for certain kinds of plants. Not all plants are mycorrhizal, and it's actually inhibitory. It's detrimental to things like the brassicas, the cole, the kale crops, the mustards, to become mycorrhizal. And some of the really interesting work by Mike and Edie Allen, for example, showing that if you've got a plant that is strongly mycorrhizal, it can send out those mycorrhizal hyphae and force colonization on something like broccoli or cauliflower, a brassica. And the brassicas should never become mycorrhizal, or not really helpful for them, doesn't benefit them. And then the mycorrhizal plant that's forced colonization will actually suck nutrients out of the non-mycorrhizal plant and cause it to die. So got to understand the mycorrhizal fungi. It's often presented like, oh, plants must be mycorrhizal. It benefits everything. Nature doesn't work that way. So you want to be very careful with your non-mycorrhizal plants that you have a lot of actinobacteria in the soil, because those actinobacteria help prevent colonization of those plants' root systems, prevents the mycorrhizal colonization, which is great. But then when you get to our mycorrhizal plants, which are, most of our carrots, our vegetables, the tomatoes, the potatoes, the solanaceae, all of our typical veggies that we like to grow, they need to be mycorrhizal. So there's where you want to not have so many actinobacteria in that soil, so they don't prevent colonization of the root systems of plants that need to be mycorrhizal. Now, here I'm talking about mostly endomycorrhizal fungal species, the VAM, vesicular arbuscular mycorrhizal fungi. When we get to plants that are evergreen, they need to be ectomycorrhizal. And so it's a whole different set of spores that you want to be putting in when you're trying to grow conifers, when you're trying to grow evergreen plants like rhododendrons or azaleas, things like that. So you need to fine-tune things a little bit more than I think comes across with many of the people who really push mycorrhizal fungi as being like the end-all and be-all. Your plants will grow really, really well. You don't need anything else. Oh, yes, you do. Because if you've got just mycorrhizal fungi, nutrient cycling is not happening. And you can't supply everything that your plant needs in terms of nutrients with just one mycorrhizal species. Some of the more interesting mycorrhizal research that's happening right now is where people are showing that each species of mycorrhizal fungus really only goes after several different mineral nutrients. There's no mycorrhizal fungus that will go out, search, and bring back all of the mineral nutrients that your plant requires. So you've got to have a root system that's putting out the different exudates to wake up the different mycorrhizal fungi, so you'll get all of the nutrients that you need, or you need to have the bacteria and fungi being eaten by protozoa, nematodes, microarthropods, earthworms, things like that, and get that all of the nutrients cycling. So you can see where we've just barely in this talk started to get into the nitty and gritty of how all these different organisms interact and function together.
Excellent. And for my own research, I found that Radical Mycology by Peter McCoy and Elaine's work together are more broad and more general and more in-depth. So it's less focused and then seems like it's applying to everything else. It gives you a more complete picture. And often with people like that, you're going to get that it depends answer, or you're going to get more complexity than you were prepared for, than the YouTube prepared you for. But that's it. That's it. That's why you have these classes, because there's more complexity. There's more to learn, So Here we go. Next question. Will compost tea and compost outperform a mix of green manure crop?
Yes. Quite often when you're putting in a green manure, and by green manure we're just talking about plant material that has not gone senescent or dormant. So that set of nutrients still contains the set of compounds in a green manure, has the proteins, the carbohydrates, and the sugars still in that plant material. So when you mix that in, if you have good biology in your soil, then that green manure is going to decompose very rapidly. It's going to get turned into all of this different kind of soil organic matter. It's going to grow a whole herd of different species of bacteria and fungi, which of course leads to a whole herd of different species of protozoa, nematodes, microarthropods, earthworms, the whole system. So the green manure is the food to feed that first stage that you have to have present in your soil to have a food web. But what if you're mixing in green manure into dirt and there is no biology present to utilize that material? Then ultimately, who's going to end up decomposing that green manure? It's going to be the disease-causing bacteria, fungi, the bad guys, the things that are actually going to do more harm than good. So you need to make certain that you've got the whole system together. I always love it when, scientists, we've got to say, what's the most important thing? Well, how can you grow microorganisms if you don't have food? Yeah, but if you don't have microorganisms, that food's not going to be used. So you got to have both. You got to have the whole system working together. I always love people who have to say, well, the foods are the most important thing. Great. So if I put a smorgasbord with the most fantastic sets of foods out in the middle of the Saudi Arabian desert, what's going to happen to those foods? There are no people. You're not going to get anything good out of a smorgasbord sitting in the middle of an area that has no life. Same thing in your soil. No life, it doesn't do any good to have green manure.
Excellent. And I also, green manure would favor more bacterial growth than fungal growth, correct? Because it's all the sugars, enzymes, right, and chlorophyll still there.
So it depends on exactly which green manure you're talking about and how, here's the it depends.
Yes, I love it.
So if, let's say you're cutting down corn stalks while they're still green, there's a lot of bacterial food in there, and so you're going to grow a fair amount of bacteria. But all that cellulose, all that lignin, all those really complex carbon compounds, that's what grows fungi. But let's say you're going to go and you're going to cut your green peas. You've harvested the peas, but now you're cutting down the green plant material. Oh, are you going to grow bacteria? So what does the plant you want to grow need? Mm-hmm. And that's why I'm usually going to recommend to people that they take all of those plant materials and turn it into compost. So you're already growing all of the right sets of organisms. Well, then you better have a recipe for your compost that's going to select for the organisms you want. Do you want more green plant material in your compost? You're going to grow a lot more bacteria. Is that what you need in your soil to grow that plant? Or do you want a whole lot more woody materials that grow fungi? I want those stalks. I want that tough woody material in there because that grows fungi. Is that what you need for your plant? So you got to learn those balances. So again, we've only barely touched the beginning of all of this.
Wonderful. Okay. I love this. Did you want to jump in? You got that next one, Kelly Shin.
Yeah, well, we got a lot of questions. How about we move to some of the really advanced questions?
Okay.
We're going to have to machine gun.
I'll try to make the answer shorter.
Yeah, so here we go. From Una Greenway, I really like this one. We have a root nematode in our coffee trees. Do we remove the trees to solve the problem? We've been organic farming for 39 years. We have lots of organic matter each year. Is there a ground cover that we can help to plant our trees?
What you're going to have to do in that particular instance is to get the beneficial nematodes back into that soil. So make a compost that has a lot of the bacterial feeders, the fungal feeders, and the predatory nematodes. And you may have to buy an inoculum of those nematodes. And we extract and sell those inocula. If you're in a different country, that's going to get a little, we might have to teach you. You might want to come up to the farm. And in about 3 days, we can teach you how to extract your own nematodes from your soils, teach you the microscope work to identify the kind of nematodes that you have. So is this, are these good guy nematodes that we want? You put them into your compost pile, you grow up billions of those kinds of really beneficial nematodes. You make a compost extract or a compost tea from that compost and you inject that into the soil around your trees. No, do not take the trees out. You want that food going into the system. If you've got plants, like often under coffee, a groundnut, a mini clover, some of the dichondra that you have in, and I know most people in tropical systems are like, oh, dichondra, it's a horrible weed. No, we're talking about the short little dichondra, not the things that get real rank and kind of try to grow over your trees. Put in those cover plants so they will maintain these really beneficial nematodes. We want to make certain that you're putting mycorrhizal spores on coffee. They have to be mycorrhizal. And mycorrhizal fungi will actively capture and consume those root-feeding nematodes. So again, a lot of biology you got to get into that level of understanding what happened that the root-feeding nematodes are taken over. But yeah, you're going to not have very good yields until you deal with that.
I really like this next question. Okay. Hi, Elaine. This is from Kirsten Huber. Thank you for the presentation. How do you handle the encroachment of glyphosate in nearly all we grow? Whether organic, biodynamically, how do we prevent and handle the promotion and promote its microbial degradation and protect ourselves from its chelating effects on our bodies? And can you share local compost soil resources for the DWF area? So,
it's going to take me about three hours to answer that one. So the quick answer is you have to get the biology back into the soil. To decompose the glyphosate, there are many different species of fungi that very rapidly and very easily decompose glyphosate. You got to get that inoculum into your compost, make an extract, make a tea, inject that into your soil so you free up all of those nutrients that are bound by the glyphosate. You've got to decompose that toxic material, and it's only by getting this food web back into the soil. Protecting yourself from further drift from your neighbors, you're going to have to put up a barrier of plants. So you want a row of trees, you want a row of shrubs that whichever way the prevailing wind is coming from, you are blocking that glyphosate drift. It's getting caught on the leaves and the surfaces of those trees and bushes, which means probably once every 3 months through the growing season, you're going to have to go out with a compost tea and spray the surfaces of those plants above ground so that it will decompose that glyphosate that is now captured on the surfaces of those leaves.
Amazing.
Yeah, I mean, we've got that problem in the farm here in California where we are still dealing with some parts of the farm where nothing grows. We've owned the farm now for about 1.5 years, and for the second growing season, we have spots that are just absolute bare soil. And it's just going to take time. We've got to make enough compost to have the excess compost to put on those areas where there is no plant life at all growing because of the Roundup that they sprayed.
Nice, great question. Okay, really quick question from Kurt Davis. Can you use a local compost source and add a compost tea to enhance it?
You can, but you're going to have to get your microscope out and look at that local source of compost. If you go to a municipal composting yard, I can almost guarantee you that's not compost. That's putrefied organic matter. That is not something you want to touch because it'll kill your plants. So how do you tell whether it's good compost? Good compost is the color of 70% cocoa chocolate. So go to the grocery store, buy a 70% cocoa chocolate bar, buy a milk chocolate bar, because there's your, this isn't good stuff. And bring along a black t-shirt or black colored something. And when you go out to that compost and they say, oh, this is our best compost. Oh, really? Have you looked at the biology? What? What? Biology? What are you talking about? Yeah, it's only what does the decomposition of all of that organic matter. But dig into the pile a little bit so you've got moist compost. And then whip out the chocolate bars. Is it the color of that milk chocolate bar? That's not decomposed. That is chock full of pathogens and pests and eggs of every kind of horrific, you don't want to, do not touch that stuff. If you buy it, you're going to have to compost it, which means, you know, letting the worms deal with it, letting the temperature get back, all of that good stuff. Take the compost course. So another thing you want to, look at the color. If it's black in color, there are no nutrients left in an available form in that material. That's not compost. And it has no life left either because they've been charcoaled. What you're buying is charcoal. And don't bother with that. You would have to add all of the biology back into it. So yes, you could buy a compost tea and apply the compost tea into that black material, but how are you going to put back the nitrogen, the phosphorus, the sulfur, all of the chelated organic compounds that should be there to feed your plant? How are you going to put that back into that putrefied organic matter? So lots of problems. You've got to have that microscope to look at that compost material and use that to tell you if you still have a decent set of biology in there at all. And most municipal composting operations, and believe me, every state in the United States, we've looked at that compost and it's just not compost. And the problem is, in the reduced waste industry, they've been trying to convince people that reduced waste is equal to or the same as compost. Absolutely not true. You make compost for the organisms. The organic matter is just a nice addition, but what you really want are the organisms.
Excellent. So here's another question, and I'm totally wanting to hear about this too. Any specific advice for incorporating these techniques with biochar?
Oh yeah, yeah. If you make biochar, and so you've got charcoal, basically. You've got woody materials that have been preserved through that process of heating it to an extremely high temperature with no oxygen in the system. So combustion doesn't happen. But all those organic compounds are now in a very much preserved form. All of the biology has been killed when they're making biochar. It's charcoal. So take that biochar and spray it with really good compost tea. And all those organisms move into the cracks and crevices and all of the condominium housing that's present in that char material. And now you have biochar. But unless they've actually done that, they've actually either mixed the char with compost or they've applied that set of microorganisms from compost to the char, it's not going to be doing much for you. Char, without the biology, is just a physical structure. So I don't quite understand how that disconnect happened when people were developing the whole concept of biochar. You can't call it bio if you have no biology in it. So don't waste your money buying something that is strictly char. You can make charcoal yourself in your barbecue pit, so why pay somebody else to do that? Add the biology by having your compost, put the biology, mix it with your char, and now you've got biochar.
Excellent. Do you think that energy-wise, just in the whole scheme of things, do you think it's wise, energy-wise, to make biochar compared to just using the natural systems?
Yeah, I think it's better to use that organic material in a compost pile or in your soil as a food resource for all these great organisms that are out there. That's how Mother Nature has done this for the last, ever since plants have existed on this planet. So why are we trying to short circuit around, we've got to store all of this carbon back into the soil. Yeah, but is that the right form? Is that the right way to do it? I've looked at a lot of the biochar calculations where, in theory, they should get more energy back from making charcoal than they use to make the charcoal. Great, in theory. But could we actually have some data from actual places that make char showing that we've got a perpetual energy machine here? I remember the original people starting the biochar hype, and they could never convince me that what they were calculating in theory was actually how their plant was working. There were always problems. There were always difficulties. The thing broke down all the time. And then the experiments that they did with biochar was to take an untilled field This half was the without biochar, untilled, and they just planted corn into it. But on this side, they tilled the biochar into the soil and then planted the corn. That's not a fair comparison. They were not demonstrating what biochar was doing. They're demonstrating that you got to till corn soil before you put the corn in, because this side was so compacted. You know, if the corn plants were like 2 or 3 feet tall in the agricultural, conventional agricultural side, whereas over here where they put the biochar, yeah, the corn was growing normally. Look out for that in scientific papers. When someone wants to sell you something, they will bias the research. So you gotta look out for that. Yeah.
What's so incredible about your work is the fact that we can test it and we can verify it, and that's it's truth. You can't change it. You can see it, and seeing is believing.
You prove it to yourself, and you don't have to take me on trust. You don't have to believe what I say. You get that microscope, you've got your own measurement tool. End of story.
Awesome. So here's a great question. What do you do about weeds when weeds are present, like bindweed that are in ground elder or couchgrass, things that are really below surface? It sounds like they're talking about weeds that are trying to tie up the soil structure.
Yeah, and Mother Nature has sent those weeds to you to do specific things. She's trying to move that soil from an extremely bacterial-dominated system and trying to develop the fungal component within that soil. So where are you on that successional system? If you're really up here, where you've got a good balance of fungi and bacteria to grow your tomatoes, bindweed won't grow. You won't have those problems. But if you're still back here where you have not set the stage to grow tomatoes, then Mother Nature is going to try to fix that soil and move it along. And her method is to send in plants that are partway along that successional sequence. So you got bindweed, you got dandelions, you got thistles, because they're trying to move it along. If you come in with a good compost, with a good set of microorganisms, you can jump from early successional to late successional grasslands overnight. And then Mother Nature won't be sending bindweed and thistles and all those other weedy plant species. So it's just Mother Nature trying to send you a message. Start learning what Mother Nature's trying to tell you by the types of weeds that are present in the system.
Yeah, and I've even heard that they can tell you what minerals are present, what minerals are lacking by just looking at the weeds. It's pretty incredible. So here's another question from Carlos Miller. They're asking about biofertilizer and how does that fit into the food soil web, because it's an anaerobic process without oxygen that stabilizes and no longer is anaerobic. It's all sterile, or maybe not sterile, maybe static is a better term.
Yeah, if they're using biology and going anaerobic, they've made a lot of really strong acids. And that's the waste compounds that are made under anaerobic conditions. And so they have put a preservative into that mix of fertilizer. But as they went anaerobic, they blew off all the nitrogen, the phosphorus, the sulfur. It's gone. Those soluble forms of those nutrients are no longer present in that biofertilizer. So what did they add back? Did they put inorganic fertilizers that are salts, and those salts are going to kill your microorganisms in your soil? Or they didn't add them back because, oh, look at all of the calcium, magnesium, sodium, iron, zinc that's still present in the soil. But that anaerobic process produced a lot of acid. So what's the pH of that biofertilizer? 4.5? 3? 2? Because for that material to appear like it's sterile, there's no carbon dioxide being produced, is because all of the organisms have been put to sleep by those strong acids. For example, if you go back to the work by Fred Woods, he would make a compost tea with just, fantastic, huge diversity, really wonderful stuff. But he wanted to put it into containers, seal it up, and put it on the shelf in the store so that it would have a shelf life of, a couple 500 years or something like that. So how can you take a really active compost tea and put it into a jug so you can seal it? If those organisms are still active and growing and producing carbon dioxide and all those gases and stuff, boom, yeah, and Kmart gets really upset when the whole inside of their store is now plastered with brown stuff. So what he did was to take really strong acids. So phosphoric acid, for example, has a pH of about 3, 2.5. And so he would very carefully start to add that acid into the compost tea. And as the concentration of that acid got high enough, pH starts to drop, the organisms would go to sleep. Except about 50% of the bacteria and fungi in soil can't respond that fast to that preservative, and they die. They don't have the dormant stages, many of them. So you started with a really fantastic set of microorganisms, but by the time you've added that acid and dropped that pH really low, which is what they're doing with these acidic, anaerobic biofertilizers, what's the bio actually left in that material? What you have are a bunch of microorganisms that are meant to grow in anaerobic conditions. Those are the spores, those are the dormant stages you have in that material. So you put that out into your soil, you dilute the strong acids by watering it, by diluting it in the soil. What's the effect going to be on your organisms? The aerobic organisms that were in your soil. You've killed them, or you've harmed them significantly. Here's where your microscope gets to be really important. What happens when you take this product that somebody's trying to sell you? Oh yeah, it works great. Look, I got pictures where you don't have that product and where you do have the product. Right. I can show you 60 billion big plant, little plant pictures that are complete and total, well, maybe not 60 billion, but 30 billion, where they've done those kinds of experiments, and that kind of hype is utilized to convince people to buy something. So you buy it, and it doesn't actually do anything. You buy something, you put it on, it actually harms. So test those kinds of things first. When somebody wants to hype you with that kind of information, okay, buy a little bit, put it on a 10-foot by 10-foot area, get that microscope out and see what the effect of adding that material to your soil under your conditions. What's it done? And so then you know whether it is worthwhile to go out and buy that product.
Awesome. Okay, so this question below I think applies to a lot of questions people are asking that have like indoor systems. They're talking about aquaponics, indoor planning. Anyway, this question's from Chad Wasserman. Can you use a fertilizer injector with compost extract to an irrigation system in a greenhouse setting? And what can you restore diverse organisms in an indoor environment, in an aquaponics environment?
Yep. So we've done a lot of work with like nutrient troughs, with float tables, with flooded systems. So I think every kind of hydroponics or aquaponics, we've actually done testing on those, and we actually have some tanks going here at the farm where we're doing more testing on that. So what you want to do is in the tank where the water is being pumped up into your troughs or into your float tables, you want to establish this really good set of organisms in that, and you want to be adding foods like humic acids that you extract from your own compost. So it becomes very inexpensive. I don't know how many of you have gone out and bought a gallon worth of humic acid extracted from Leonardite, but it's like $50 a gallon. And you're going to have to be, in a 500-gallon tank, you're probably putting, that whole gallon of humic acid into that tank on a weekly basis. That's kind of spendy. Whereas if you make your own compost, or you have a neighbor that you can buy the compost from, you check it with your microscope, it's good stuff, you can extract the humic acid out of that material. Once you've extracted that soluble humic acid, you put the compost back in the pile, and the organisms reestablish and rebuild the humic acid. So next week you just extract the humic acid that's developed in that week. So it's a very long-term source of the foods that you put into the water. You've got the organisms in that water, so now it's pumped up and through. As it goes through the trough, or as it goes through the rock wool, or whatever your support matrix is, the organisms get established on the root systems of your hydroponic or your aquaponic plants. And now remember, the plant is putting out exudates to grow exactly the set of organisms that it requires to pull nutrients out of your vermiculite, out of your rock wool, out of your sand, your silt, your clay. If you've got a little soil start, you put your seed into the little planting tray, and you've got this little bit of soil sitting in there. Well, those organisms can pull the nutrients out. Actually, if you look at about 1 gram of soil, if you actually got the microorganisms to decompose all of the sand, the silt, the clay, the rocks and pebbles, everything that's in that little bit, you could grow a whole entire field, an acre of plant material. Now, the microorganisms are generally going to take a little bit from here and a little bit from there and a little bit from some other place, so you don't lose all, but 1 gram would grow an acre worth of most of our crop plants. That's the kind of power that's in there. And so when people want to worry about, well, this plant's going to, pull these nutrients out of the soil, all plants pull the nutrients out of the soil. They have to have the mechanism for doing that, or how could they stay alive? If every single cell in that plant has to have all of the, how many nutrients does a plant have to have? It's got to have everything. There's got to be silver in a plant. That's never included as one of those essential nutrients. Arsenic. Does your plant need arsenic? Yes, it does, at real low levels. But it's got to have them all. So if your plant is looking stressed, is it because it's not getting enough arsenic? Or is it that it's not getting enough zinc or cadmium or calcium? So I get around all of that kind of question by saying, just get all the organisms in there. So when your plant puts out the exudate saying, I need arsenic, the microorganisms are there to make the enzymes to pull those nutrients. So in a hydroponic system, the bacteria and fungi are growing on that root system. Where is the source of all the nutrients that your plant needs? Humic acid floating by. The bacteria, the fungi are going to grab onto that humic acid, pull the cadmium, pull the silver, pull the arsenic, pull the whatever it is your plant needs. And however tiny amount or large amount. Nitrogen, there's a huge amount of nitrogen present in a humic acid. But again, if you do a soil chemistry assessment, you send humic acid into a soil chemistry lab, it will come back and say there's no soluble nitrogen here. You're going to have to add inorganic fertilizer. You're going to have to add a nitrogen nitrate or ammonium to supply enough nitrogen. They're lying to you. There is more than enough nitrogen in humic acid. In the structure of that molecule, there's all the nitrogen that your plant requires. But you better have the bacteria and fungi to make the enzymes, as directed by your plant, to pull those nutrients out, whatever nutrient. So as long as you've got humic acid floating by, or other sources of organic matter, fulvic acid, oleic acid, those other complex forms of organic matter, you've got all the nutrients you need. You have to have the biology. Okay, so your bacteria and fungi are making the enzymes to pull those nutrients from the humic or fulvic or oleic acids. What else do you have to have in that hydroponic system to get the nutrients out of the bacteria and fungi? You gotta have protozoa or nematodes or microarthropods. They are all organisms that live in the water. They all require a good amount of water on the surfaces of the sand, silts, and clays so that they will go eat your bacteria and fungi, release the excess nutrients that are in your bacteria and fungi, and then your plant has all the nutrition that it requires. So all we really need in hydroponic systems is, again, to have that compost with all these organisms, inoculate the full diversity of all those organisms into your system, and then just make certain that you continue to feed the organisms with that humic acid or fulvic or whatever form of organic material that actually contains all of the nutrients that your plant's going to require.
That was awesome. So I'm scanning through. You're answering so many of these questions with your detailed answers that it's. I hope everyone's paying attention because we're getting a lot of answers. Raleigh, do you want to take over?
Yeah, sure. I mean, this is so cool. We've had 450 people stick around for 2.5 hours. You guys are champions, and it's tough for us sometimes. We can't answer all the questions of 450 people. I'm blown away by the response here. So another question we're getting from a lot of people, and I've seen like at least 5 or 6 questions on sourcing compost. How do you source quality compost that isn't, full of weeds, that will promote your soil like the one you did on the Bush Library?
You're going to have to use a microscope. Well, let's back up a step. I was talking about when you go out to look at the compost, bring your chocolate bars with you. If it's not that 70% cocoa chocolate color, don't buy it. It is going to be more headache than you possibly want to deal with. Don't buy it. If you buy it, that means you got to bring it home, and now you got to fix it. You've got to find a source of really good compost. You might have to, give us a buzz here at the Soil Food Web or at the Environmental Celebration or Nature Solution. Give us a call, send us an email. Where do you live? And we will direct you to the closest source of really good compost that we know about. Well, Matt can do the same thing. I think you make compost, Matt, right? So
I do.
Do you make enough to supply other people, though?
No, that's the thing, is most people are doing it themselves for themselves. I know that Malibu Compost has sent samples to you specifically to get feedback, and I think that if they're tied in and they know what's going on, most of these places are going to be able to answer the questions that you would ask that we're going to ask now.
So you want to make certain that you've got at least 300 micrograms of bacteria and a good diversity of bacteria. You want at least 300 micrograms of fungal hyphae and a huge diversity of the different beneficial fungal species present. You want at least 10,000 protozoa per gram. Everything I'm talking about is per teaspoon, basically. And then you want the nematodes, the bacterial-feeding nematodes, fungal-feeding nematodes, predatory nematodes. So that's a good compost. That's what you're looking for. And again, by just taking a teaspoon of your compost that you're asking, is this good compost? Grab a teaspoon of it, bring it home with you, mix it in with 4 teaspoons of water. Yeah, just little plastic bags that all, just while you're looking at while you got your chocolate bars up there and you're comparing, grab a little of the compost and stick it in a little plastic bag. Bring it home with you. 1 teaspoon. Mix 4 teaspoons of water and make sure that it's good drinking water. It's not got chlorine and chloramine in that water. Shake the mix of 1 teaspoon of compost with the 4 teaspoons of water. Take an eyedropper, put a drop on your microscope slide, cover slip on top, microscope on the stage, turn the lamp on, look through there, and right, typically that very first field you can go, wow, or you can go, oh, this isn't, no, don't buy this stuff. It's usually that simple. You might have to take the microscope course to learn what all of the different organisms are. You know, do you have to know their names? Do you have to identify them to genus or species? No. There's certain characteristics that we're looking for with each of those groups that tells you these are the good guys. That's what you want to see. If you're not seeing that, don't buy it. It's not worth your money and your headache trying to deal with it. So if you send us questions about who's the local source of really good compost, and we work with a lot of guys in Texas, so that Texas Compost Company, fantastic worm compost. Whoa, really good stuff. And John Ferguson's Nature's Way, really good compost. That's why that the Bush Library project was so successful. We had people making good compost. We could just buy it from them. If we had had to make the compost ourselves, we would have had to ask for another year on the project. We would have gotten done in 2 years instead of 1. Well, the Bush family wanted 1 year. Okay, so we managed to do it. New York City, there's a number of really good composters up there. Long Island compost, don't. It's not compost. So that's where you want to find out what you can from us. As you get into other countries, we've worked in South Africa, we've worked in Australia, we've worked in Japan, and so often we do have places we can recommend that has in the past made good compost and probably still are, and you'll just enhance the reputation. You'll get people to start paying more attention. If the composting operation you go to, you absolutely want them to do a soil food web or soil biology test so that you know what's in their compost. And you're not buying it unless they can give you that kind of data.
Nice. Okay, so we got a question from Kelly Shin that applies to a whole bunch of other questions about compaction and I guess injecting your soil biology compost tea deep enough. So she asked, what do I need to purchase if I want to inject biology into the compacted layers of my soil? We, too, are building on an old parking lot and a sheet mulch for 1 season. Good question.
Yep. Yeah. What you want, first step is usually to find out if you have compaction layers. Well, on an old parking lot, without doubt, they have compaction layers. And you're going to want to do this kind of testing every 3 months through the whole entire year, because you get different compaction layers showing up when the soil is very wet as compared to when the soil is really dry. So you want to find them all. You want to know about all the different compaction layers. Where are they, and when do they appear through the course of the year? The most horrific compaction layers are going to always show up no matter how wet the soil is. But as the soil starts to dry down, you're going to discover a whole bunch more layers of compaction. So you're going to want to get a penetrometer. Usually like a Dickie John, that's a company, $150 for a penetrometer. Or you can buy your own. You can make it yourself. You want to get a metal rod that's about 4 feet long. If you can get the hardware store to sharpen it on one end, they just bevel it into a point. So that's going to be the end that you're going to push into the soil. And you want the diameter on that pipe, probably about close to 1/2 inch, so you can push it through some really nasty compacted things. And then you go to the hardware store and you get a pressure gauge. So if that's, it's like just a little cylinder. The pressure gauge is usually on the top, and it just has a needle that points to pounds per square inch. How much pressure do you have to push that metal rod to get it to go through the soil? And so those are your readings off of that pressure gauge. But you got to buy a pressure gauge that the pressure plate is on the bottom, because that's where you're going to put your metal rod. You want it to push against that pressure plate on the bottom of that pressure gauge. So you go out into the soil, you put the metal rod on the surface, that pointy end goes on the surface of your soil. You put your pressure gauge on top of that metal rod, and you start to push. You push on the pressure gauge. So you can read what's the pressure that you've got to apply to push that metal rod through the soil. And as you start to push, you can see where it gets harder to push, harder and harder. If that pressure gauge goes above 150 pounds per square inch, most root systems of most plants cannot push their root system through something that is more compacted than that. 150, there's a normal limit. This work has been done by Pennsylvania State University, Penn State, and they have many publications on that about what are the limits for most plants. So if you've tilled your soil, if you've walked over your soil, if you've had a herd of goats, dogs, whatever you want to talk about, walk over your soil and you don't have adequate biology, you're going to be compacted about 4 to 6 inches. So as you push that metal rod, trying to get into the picture, as you push that metal rod down into the soil, you'll hit that compaction layer at about 4 to 6 inches. And it's going to take more than 150 pounds per square inch pressure to get that metal rod to go deeper than that compaction layer. How thick is your compaction layer? So how long do I have to continue pushing so hard? 200, 250 pounds per square inch pressure to push that metal rod through the soil. Typically, if there's been any big equipment that's gone across your property, and please remember as homeowners, by law, they had to compact your soil to at least 600 psi in order to put the foundation in. So are you dealing with a compaction layer in your soil? Yes. If they run big equipment across your soil in order to flatten it in that parking lot, how much effort did they have to put in to compact that parking lot enough that you could park on there under all kinds of conditions, wet, dry, frozen, whatever. It's really nasty. So you're going to have to inject things into that soil. And so an injector machine, now you know how far down. Is it at 4 to 6 inches? Do you have another compaction layer at 18, another at 24 inches, another at 3, another at 4 feet? Find out. Because you want to get in there with your injector. Just go to a regular, dang it, there we go. I have a phone call that somebody's trying to send, sorry. So you go to a box store, just your average general everyday box store, and buy an injector. They cost maybe $50, $100. The really fancy ones where you can change, the pressure of the liquid being blown out, the tip of the pipe, might be $150. So buy the one you like. And you fill up your tank with your compost tea or your compost extract. You push the metal rod through the soil down as far as you can get without breaking your injector pipe. So then when you depress the handle on your injector, it pumps that water down the pipe, and the hole that allows that compost extract or compost tea, the biology, to move out into the soil is at the tip. So you push that pipe all the way down as far as you can go with it, start pumping the compost, the organisms out into that soil, and pull your injector all the way up out of the soil while you're still pumping the compost tea. That means that whole entire column of soil, you've just injected biology out in every single direction. That whole column of soil, that good half foot, 3/4 of a foot, is now been inoculated with that really good biology. And you've gone up through all of the layers of compaction. Hopefully, down at the bottom of that hole, you've got biology that went out below that. And so you're maybe going to improve structure in the top 5 to 6 feet of your soil. And now you've got biology down there doing its thing, happily making it better. And that just keeps growing out further and further and further. If you have pesticides down there, you might have to do injection a couple of times, because sometimes the first wave of colonists don't quite make it. So you got to put the second wave of colonists in. Think about the Pilgrims going to Plymouth Rock. The first wave of settlers, 90% of them died. The second wave of those settlers, 50% of them died. The third wave of settlers, only 10% died. And therefore we have the United States, because that was the colonization wave that really started it all. Thank goodness there were 3 waves of pilgrims to come along and start the colonization. Many other waves of colonization after that. So we've got the great diversity that we have in the United States. So we got to do that everywhere on the planet.
Nice. So many good questions coming in. We'll probably group them into a few really good summaries of all the questions, but just in case if you feel like you didn't get enough information about the course, check out the handouts, because we have the information about the right link, and we'll send you the replay and a link to it afterwards, just so you can check it out if you want more information, because you can jump down the rabbit hole and get so much out of this. I know I've had a great time so far, and I could stay on here forever. And just as a reminder, that offer expires on Monday. So for people who want to take Elaine up on her amazing offer. So here is another grouping of questions. It's about the balance of perennials and invasives. So a lot of people are asking about how to keep, weedy grass out and promote perennials. So J. Scott Anderson asks, in perennial grass forage stands, how does one overcome invasive or annual overgrowth such as ragweed?
You're going to have to get that soil more and more and more fungal. So you select against the ragweed. When you start to understand what bacteria do with soluble nutrients in soil versus what fungi do with soluble forms of nutrients. Oh boy, we're getting into chemistry now. Look out. Try to make this simple for you. Bacteria and fungi do very different things. Bacteria, when they grow, produce a lot of glues around their body. So think about this little tiny bacterium. The end of my finger is a little bacterium, 1 micrometer by 1 micrometer in diameter. And that bacterium, in order to stay where he's going to get food, he's putting a whole bunch of glue out. The glues that bacteria make are very alkaline. So any aerobic bacteria, not anaerobic bacteria, they're different. But the good guy bacteria that we want to have in our soils produce these glue layers, and all of those glues are on the alkaline side. So a bacterial-dominated soil is going to have an alkaline pH. All of the Great Plains of the United States, where we've got good, healthy grass production, they're alkaline. Go out and measure the pH. They're somewhere between like 8 and 12. And people often think, oh, you can't grow plants in a pH of 11 or 12. Oh, yes, you can. Go to the Great Plains and start measuring pH. There's plenty of places that have a pH very alkaline. So the bacteria growing, they're what are producing that. So you might suspect in those early successional grass species that are strictly bacterial, we've got very high pHs. As we start to get a little bit of fungi into the system, well, fungi don't make the glues. They don't make the amount of glues that they make a little, but they don't make the amount of glues, nor are their glues extremely alkaline. Instead, fungi produce organic acids. Their waste compounds that they pump out of their bodies are organic acids that have a pH between 5.5 and 7. So if you have a fungal-dominated soil, the pH is going to be somewhere between 5.5 and 7. It's going to be on the slightly acidic side, which is great for our later successional plants. That's what they need. This difference in the pH that the bacteria or the fungi are imposing on the soil have a consequence for the forms that nutrients exist. Most of the research has been done on nitrogen, because when bacteria and fungi are eaten by protozoa, nematodes, microarthropods, earthworms, the whole slew of predators in the soil, the form that the nitrogen is released is NH4. NH4, ammonium, only continues to exist in that form is when it's in an acidic soil. So when we're dealing with fungi being predominant, or where we're dealing with spots where the fungi are predominant, and maybe you've got bacteria around there, and you're looking, another phone call tried to come in, sorry. When you've got fungi, maybe spots of fungi, spots of bacteria. So may have a lot of bacteria, or fungi. So a good mix when we've got those kinds of balances. Where the fungi are, they've reduced the pH down to less than 7. So the ammonium, when it's produced, released from bacteria and fungi being eaten by their predators, that NH4 stays as NH4. And that selects for perennial plants. That is the form of nitrogen that is best for perennial plants. Whereas if we've got a lot of bacteria, the pH of the soil is going to be alkaline. When that NH4 is released, when the bacteria and fungi are being eaten by their predators, that NH4 is going to be almost instantly converted to nitrate and nitrite. Because only under alkaline conditions will the nitrifying bacteria produce the enzymes to rip the hydrogen off that NH4, ammonium, and stick oxygen onto those nitrogen molecules. So you end up with NO2 and then eventually NO3. Nitrifying bacteria get energy from doing that. But that's why alkaline soils bacterial-dominated soils, tend to be strictly nitrate present in that soil, which is perfect for growing weeds. All of our weedy species, all of those things that all of us gardeners, all of us agricultural people hate because they're trying to steal nutrients away from our plant. Oh, but you know, if you've got really good nutrient cycling in your soil, what does it matter if you got weeds? Because in the root system of your crops, you got good nutrient cycling going in there, and you got weeds, roots over there, who cares? Your crop plant is still getting all the nutrient that it requires. Well, it looks messy, it's taking water away. It's okay, so make sure you've got the balance of fungi and bacteria right for the crop that you're trying to grow. Make certain that you don't have the balance of fungi to bacteria that's going to grow the weedy species. So where in succession do you find your least favorite weedy plant? And make sure that you don't have that balance. So, in some of the books that I've published, 10 Steps to Gardening with Nature, we have a list of a lot of those plants. I don't always have every species known to man. So you might want to send me an email if you take the classes. I'm very open to answering questions for people who are paying money to help me learn more about these kinds of issues. What is the fungal bacterial biomass ratio that's going to select for which perennial weedy species are you considering? Which annual weedy species are you interested in? And so we shift away from what selects for them. Most annual weeds are not mycorrhizal. So get the mycorrhizal colonization on the root systems of your crop plants. If that's the kind of crop you're growing, make sure you've got the mycorrhizal fungi in there. And that all by itself will typically select against the weedy species from being able to grow.
Awesome. Good question. Okay. So we're going to close this out in, I think, 10 minutes. Elaine, you've been a champion for sticking with us for 3 hours straight. I know you
I have no problem. I could keep going.
Okay. Well, I'm going to say, Matt, if you got to get going, I understand. He's got a family. I don't. I'm a crazy bachelor who's got nothing to do but answer people's questions on webinars, so I'm totally down to do that. So next, a big business question. So this is a grouping around business and doing this on large-scale farms. So a question from Zach Lewis. I have a question regarding application on large-scale farms. I think it could be huge if we get it in the hands of some big-time investors. So his question is about basically converting large-scale farms, restoring the biology, and can you, make it profitable through that? So he's saying, let's say can you reduce expenses on a large-scale farm by restoring the soil biology? You know, is it affordable to do this, and how much are they going to see their bottom line increase?
So we've worked with a number of dairies where, you know, they came to us because they were having to spend so much money on the fertilizers. They had a lot of sickness in their animals. They were all diseased. They all had drippy, runny noses. They had lots of problems with birthing calves, and they wanted to know if there was anything we could help them with. So we took all of their waste materials, turned it into compost. What we did was to send the turner out to their farm, and we set up routes where for these 15 days, we would be there with the turner whenever it was needed. And because of the recipe, the compost was exactly the same. We knew exactly which days we needed to be there. So they took all of their waste material, turned it into really good compost, spread that out on their soil. And in the first year, they no longer had to buy any inorganic fertilizer. They did not need any pesticides. They had lots of problems with a number of different insect pests. So they got rid of the whiteflies, they got rid of the aphids, all of the various kinds of leaf hoppers and things that would grow on their alfalfa, that would chew up their legume stands. Gone. They did not have to buy those pesticides anymore. No more fertilizers. The plants, as soon as they put that biology on, we doubled or tripled the rate of growth of the grass. Clover came back into their fields. I remember when the first agronomist went out to their fields and saw that clover, the agronomist went crazy. Oh my God, look at all the clover in here. If your animals are in here eating all that clover, they're gonna die from bloat. You've got to get out there with the herbicides and get rid of that clover. And all of the dairy guys said, we have absolutely no bloat in any of our animals. We've gotten rid of that problem ever since we went off the inorganic fertilizers. Our animals have been out here eating these pastures that are 50% clover for the last 3 months, and we have no bloat. As long as you're not using inorganic fertilizers, you don't have that problem. So a lot of discoveries like that. Working with most of these dairy people, on average, so here's the average savings on a 300-acre dairy farm of $200,000 reduced expenses. That's including the reduction in what they're paying for water, because we have reduced their water use by 30 to 50%. No more inorganic fertilizers, no more pesticides. The veterinary bills went way down. They used to have the vet come out probably once every 2 weeks for bloat, for calving problems, for myriad different problems. The vet came and visited once a year to certify the herd. That was it. How much would you reduce your fees just by that? So all of these benefits, if you go to the website camperdowncompost.com, it's a company that we quantified all of this work in Australia. They have over 275 clients. All dairy farms that they're working with, they're expanding. Geelong composting is another aspect. That's working with the vegetable farmers, and they deal with huge acreages. We worked in South Africa. We're at the tomato farm. That when we started working with them, they had 300,000 hectares of land. In order to have 5,000 hectares that they could actually grow tomatoes in on an annual basis. Once they started working with us and they could now grow continuous tomato year after year after year, they did not have to rotate. So they started expanding the crops that they grow. They now grow onions, they grow avocado, they grow a whole number of different crops because they got 300,000 hectares of land. What do you do with it when you don't need all of that? They have 50,000 hectares to grow tomatoes. So all of their land, well, they put in an elephant farm is another one of the uses that they put that land to. So they take herd and difficult elephants, and they put them back out in the wild, basically on their own property that they're not using to grow tomatoes anymore. So can we do this large scale? Absolutely. First thing you got to learn to do is make good compost. And that seems to be a really hard problem for most people, that they can't follow directions. Um, Come up to the farm. We have a windrow turner. We make compost in windrows. We make small piles of compost. So it's a good place for you to come for the practical hands-on. We require that you take the online courses. And you probably want to realize that when you sign up for the classes, we give you a password that lasts for a whole entire year. You can go back and review the lectures over and over and over again. I remember one meeting that I went to not that long ago where one of the people in the audience came up to me and said, "You know what, Dr. Elaine, you sound exactly like you do in the online courses." Phew, good thing. I've listened to you, each one of your lectures, over 54 times. And every time I listen to you, I put together more information. I make connections. Oh, if I'm seeing it, that means that. And so then I don't have to, and I can do, oh, wow. So every single time. And for those people who need to hear it over and over again, we give you that ability to go back and listen to it again and again and again. Because sometimes that's just so chock full of information. Like, how many of you are going to remember absolutely everything I went over during this 2.5 hour period? It's really nice to have the ability to get this whole session and be able to listen to it again and again and again. And that's what we do in the online courses. It's go back and listen. Go back. Well, Elaine said something about that, but I don't remember exactly what. So go back and listen to it again. Nobody's going to know that you listen to it over and over again, so that you need.
Awesome. Okay,
All right, well, I'm going to take off. Thank you so much, Elaine.
Sure.
Being a champion, Matt. Thanks for sticking around so long.
Yep. We probably should bring this to a close. Probably everybody else has other things that they need to go do today as well. And I have people here that are looking at the watch going,
breaking down the doors.
yeah. So thank you so much for inviting me to come on and give this talk and spread this knowledge. Thank you, Matt, so much for this recommendation. It's been fantastic.
And thank you, Elaine, for being on.
Yeah, thank you so much.
All right, everybody. So this was a replay. Some people asked me for the website. So see the email that came to you. It has a link to her website. Just look for the email with the invitation to this webinar series. You'll find it. Uh, and also come back next week. We'll have another session with Elaine. So thank you so much for joining, and